edition of Horizons in Molecular Biology. It is my great pleasure to be opening today's special edition, this year's special edition. So first off, I would like to introduce a bit the concept of this conference. The Horizons in Molecular Biology is organized every year in September by the students of the International Max Planck Research School for Molecular Biology in Göttingen. It started as a small symposium, but it has grown largely over the last years. And for the past 16 years, the aim has always been to bridge a gap between young scientists and experienced researchers from academia and industry. Science is supposed to be interactive, and the aim of the venue has always been to create the perfect atmosphere for peer-to-peer interactions, as well as knowledge transfer. The current challenging situation struck us all, but we took the challenge and decided to go online with Horizons for the first time in 17 years. This, of course, is quite fascinating at the same time, since it allows us to reach out to even more researchers around the globe. And this is proven by the fact that so far we have got around 800 registrations from many diverse countries. And so this is how our conference looks this year. And we have, so basically since the conference started in 2003, many scientists have passed by the City of Science, Göttingen. And this year we have the pleasure of having speakers from many different fields, such as molecular genetics, developmental biology, structural biology, cell biology, bioinformatics, evolutionary biology, biophysics, and membrane biology. Now, this is how the schedule looks for the rest of 3 days of the conference. And I would encourage everyone to not miss our keynote talk, which will basically happen after this opening ceremony, which is given by Richard Henderson. And we also have other exciting scientific sections happening today, which you should not miss. And this is also followed by our awarded student talks, poster sessions, and panel discussion. Our panel discussion will take place today at 5 p.m. before our second poster session, which is at basically 6:30. And the poster session will be hosted in Zoom. And besides, I would actually like to remark that we have a social event coming up, which is open for everyone. And it will happen tomorrow, Wednesday, 16th of September at 8 p.m., which is called Join Us for Horizon Sandrates. So the idea is that you join us for discussions with any refreshment that you would like to join us with. And also, I would like to take this opportunity to ask you to share pictures either of from where you're watching the conference, or just a picture of yourself as well as maybe the conference in the background with us in Twitter, just sharing it with the hashtag Horizons 2020 so that we can have all more a feeling that we are interacting with each other and it doesn't feel so empty. All right, so since this year Horizons takes place online, I would like briefly to introduce you or to get you familiar with Big Marker. So on the left-hand side, you can basically see the landing page of the conference. This is the place where you can navigate through and see all the talks that are coming up. And basically, if you just click on one of them, you will be redirected to another page, and you just simply need to press on Enter Webinar, and you will be just at the talk already. And this basically brings us to the right-hand picture, which basically is the webinar room. And here there are many functions that you should know, but I think the most important ones is that if you want to ask the questions to the speaker, please use the Q&A section on the right side of the chat. And for any other related discussions, please use the chat panel. And lastly, the biggest thank you goes to the organizing team of Horizons 2020, without whose input and incredible efforts, it would have been impossible to put everything together and have you here today. We would also like to thank Stefan Burkhardt and Kerstin Gunige from the IMPRS Molecular Biology Coordination Office. Their years-long experience, networks, tips, and advice are key every year in organizing a successful Horizons. Also, we would like to take this opportunity to thank the NPI VPC Media Service, especially Irene Bokche-Gajewski, who took pictures of the group before Corona times, and Johannes Poli, who helped us enormously with setting up the online conference booklet and our promo video. We would also like to thank our sponsors, the Georg August University of Göttingen, as well as the Max Planck Institute for Biophysical Chemistry, for the support on our conference. And last but not least, we want to thank all the participants and speakers who will join and have joined the Horizon Symposium so far. And all the work would not be complete without the ideas, talks, and questions brought from all over the world in order to make Horizons a fun scientific journey. And now, since I know you're all waiting for it, I would like to hand over the session to my colleague Valik, who will introduce the keynote speaker of Horizons this year, who is Richard Henderson.
Good morning, everyone. Today, it's my pleasure to introduce the keynote speaker of Horizons 2020, Professor Dr. Richard Henderson. Richard Henderson is a structural biologist with a physics background from Edinburgh University. And after a PhD at LMB working on enzyme mechanics, he developed an interest in membrane proteins as postdoc at Yale. Back at LMB with Nigel Unwin, he used electron microscopy to determine the structure of bacterial rhodopsin in two-dimensional crystals, first at low resolution and later at atomic resolutions. With Chris Tail, he helped to develop the method of conformational thermostabilization that allows any membrane protein to be made more stable, while at the same time remaining a chosen conformation of interest. This helped the crystallization and the structure determination of several GPCRs and led to a founding of the MRC startup company Heptase in 2007, which is now wholly owned by Japanese company Sosai, developing new drugs for medically important GPCRs. For the last 20 years, Richard has been working to improve the methodology of single-particle cryo-EM, which has recently reached the stage where it's possible to obtain high-resolution structures of wide variety of macromolecular complexes routinely without crystals. For developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution, he was awarded with a Nobel Prize in Chemistry in 2017, jointly with Jacques Dubouchet and Joachim Frank. His research is now focused on solving the remaining problems in cryo-EM so the method can reach its theoretical potential. Now I would like to invite you, Mr. Henderson, to our virtual stage. So that would take a moment to make a stable connection. Okay, great. So
you can see me and hear me now, can you?
Yes, we can.
And my screen, shall I put my, I can put my...
Yeah, you can just now share your presentation and it will be...
Oops, sorry. That was... So can you see the title slide now?
Yes, perfect.
So are we ready to start?
When you want.
Okay. So it's a great pleasure to join you all. I don't know how many of you there are, but the difficulty with web talks is that the speaker doesn't really have much feedback. So I'm just going to assume that all is well, and either Valentin or Giulio can jump in if there's any problem and tell me what the story is. So I have a talk today to tell you a bit about the impact that electron cryomicroscopy, cryo-EM we call it, is making in structural biology. But the organizers encouraged me, and probably the other speakers, to give a sort of broad introduction first. So I have about 10 or 12 slides giving you a background in structural biology, and then we'll switch over and focus more specifically on cryo-EM and what's been done with cryo-EM, how well it's doing, and what the looking forward, what the future holds. So structural biology didn't really exist sort of 50 or 60 years ago, but there are 4 general methods that have had a lot of impact at each point in time as the power of the structural biology approach has been developed. So when there was no X-ray crystallography, no electron microscopy, people did model building. Then in the sort of 1950s, 1960s, X-ray crystallography came in, starting with the atomic structure of myoglobin back in 1959. I'll show a little bit about that. Then there was a period probably of about 20 years when certain proteins couldn't be crystallized, and NMR made a big contribution, and probably about at one point at its peak, probably a quarter or a third of new protein structures being determined were done by NMR. And then more recently, electron microscopy and cryomicroscopy particularly has come into being able to deliver on the promise that it always had. And the protein that I worked on, which made me make the transition from being an X-ray crystallographer as a PhD student to being an electron microscopist, came with the protein bacteriorhodopsin. And I'm now going to put on the laser pointer here. So that's a little bit about the background of structural biology. I'll just show 1 or 2 slides about each of those methods, and then the scene will be set for getting into cryo-EM. So the first slide is a picture of Linus Pauling, who was awarded the Nobel Prize in Chemistry in 1954 for, among many other things, in working on the structure of atoms and crystals. He and Linus Pauling and Corey were the ones who built models. Initially, this is the alpha helix that forms the core of many protein structures, and the beta sheet structure. And this is him with his model pointing at it. Another very important step in the history of structural biology came with the work on DNA, and on the top left is a picture of the B form, the hydrated form of DNA, taken by Rosalind Franklin's student, Raymond Gosling, and that was the source of the early data that provided the experimental basis that allowed Watson and Crick to piece together information from lots of other sources and propose the double helix structure of DNA. So the alpha helix and the DNA double helix were the early beginnings of structural biology. So then if we switch to the beginnings of the experimental approach, this is a picture of sperm whale myoglobin from the early work of John Kendrew, who, along with Max Perutz working on hemoglobin, led a group in Cambridge to tackle the determination of experimental structures of proteins that at that point very little was known about it. And it took them a long time. They tried a lot of different methods, but eventually they got isomorphous replacement with heavy atoms to determine the phases. And this was the early model at 5.5 angstroms, made out of plasticine, Play-Doh, showing sausage shapes like this and these are 10 angstroms apart, and these turned out to be the experimental observation of the same alpha helix that Linus Pauling had proposed based on his theoretical studies of small molecule crystals. And the disk in the top right is the heme group that gives the myoglobin and hemoglobin its characteristic color. And then a couple of years later, they were able to get, in 1959, the first atomic structure of any protein. And this is myoglobin. Again, here is the heme group. But now every atom can be seen. And this is a famous watercolor painting, because it was before computer graphics, showing all the atoms of myoglobin published in the center page pullout of the Scientific American article. So that was a little bit about the background of X-ray. And then the other method, NMR, nuclear magnetic resonance spectroscopy, Kurt Wuthrich was awarded Nobel Prize in Chemistry 2002 for developing the power of the spectroscopic indirect method of determining protein structures. And this is Kurt on the top left here. And these are a structure and the experimental data behind it of a CD59 cell surface protein done in the group of David Neuhaus here at MRC LNB in Cambridge. And then coming to the beginnings of three-dimensional structure determination using electron microscopy, there were a few groups in Europe. Aaron Klug's group here in Cambridge. There was Walter Hoppe in Martinsried, Ernie van Bruggen in Denmark, and a group in Switzerland that Dubochet had worked at when he was a student. And what they showed is that from these 2D images on the left of T4 phage, and this is the phage tail, you could, knowing that that was a projection structure of the negative stain of that structure, end up with a 3D structure in the panels on the right-hand side. So that's a kind of a bit of a background to all of structural biology and where we got to. So what about cryo-EM? And so this is a slide that Tim Baker and I published in 2001 in the International Tables for Crystallography, Volume F, which is still, it's been updated and so on. And our idea was to try and show the then dominant practitioners of structural biology, the X-ray crystallographers, to describe what cryo-EM consisted of. And this is still a very good way of introducing it. In this volume, there were about 80 or 90 chapters, but there were only 2 of them that were about using electrons. One was on electron diffraction, written by Hua Chu, and then this one on cryo-EM imaging that was written by us for crystallographers. And so the idea is, and so each of these 4 different panels represents a different flavor of electron cryomicroscopy applied to different types of specimens. So on the left is a picture of E. coli 70S ribosomes from the group of Joachim Frank, who had worked on ribosomes for 20 years before that. But by the year 2001, they'd reached a resolution of about 11 angstroms. It allowed them to see the small subunit, the large subunit, tRNA, and some longer stretches of RNA double helix. And that was single particles without any symmetry. The second panel along is single particles randomly distributed on the electron microscopy grids with symmetry of, in this case, icosahedral of a virus particle. This is from Bettina Botcher's work when she was a postdoc in Cambridge. And these hepatitis B cores have 240 subunits. So 4 subunits per each of the 60 icosahedral asymmetric units. And this was the structure that she got with recording images on film in those days. And these protrusions have little ridges along the side. It was at about 7 angstrom resolution. And this is a little 4 alpha helix bundle, same helices that Pauling and then myoglobin, protruding from the surface of the virus. And this was the first time that any single particle electron cryomicroscopy reached a resolution that was below 10 angstroms, below 1 nanometer, so sub-nanometer resolution. And then the last two slides, one is a biological structure in the form of a helix. So this is thin filaments from muscle with the head group of myosin attached to it. So they're decorated thin filaments. And this is from the work of Ron Milligan's group at Scripps in California. And by doing plus and minus structure determinations, which is the illustration at the bottom, they were able to show that these decorated thin filaments consisted of a core of actin, the blue, with tropomyosin in green, and then the various different subunits of the myosin heads that were attached to the actin, simulating the contractile stroke of muscle. And even at 30 angstroms resolution, it told you a lot about how the biology was working. Of course, now these have all been taken to much higher resolution. But the final slide is an image of a 2D crystal, and you can't really see anything in the image now because the crystal is very, very thin and doesn't have very much contrast. The modulation you see is just from the spot scan type of imaging that was done. But processing these images allowed Werner Kuhlbrandt, when he was a group leader at the EMBL and before he moved to Frankfurt as a Max Planck director, to determine the structure of this light harvesting complex number 2, which is the protein structure with 13 or 14 chlorophylls in it that makes up most of the green that you see when you look outside and you see the grass and the trees, LHC2. So that was a spectrum from single particles without symmetry, single particles with symmetry, and one-dimensional and 2D crystals. And when we were writing this in 2001, electron cryotomography hadn't made much impact at that stage. It was in its very early stages, but we would now, if we were making this again, we'd add a fifth panel showing that by taking multiple orientation images of some single region of a specimen, you can, by tomography, determine the structure or observe the structure of almost any type of specimen. And that's now increasingly having a big impact, but I won't talk about that today. Okay, so that was how we described electron cryo-EM in the year 2000. But my background was in X-ray crystallography originally. And when I first made the transition from being an X-ray crystallographer to being an electron microscopist, it was via working on two-dimensional crystals with Nigel Unwin. And this is us probably about 20 years ago. And what we worked on was this bacteria rhodopsin molecule from the membranes of halobacteria. This is a freeze fracture picture taken by Walter Stekenius, who discovered these different membranes. And these little patches here have one membrane protein in them, and they are in the form of little 2D crystals. So this was a very good way of getting going with the power of electron microscopy to determine biological structures. The rest of the membranes have lots of other different proteins and are sort of randomly distributed. This is a freeze fracture picture. So by isolating these patches, you can get an image like this on a amorphous carbon grid, and each of these circular patches, or oval or spherical or slightly misshapen, is a single crystal with maybe 10, 20,000 molecules all in the same orientation. So Nigel and I were able to take electron diffraction patterns and images. This is the calculated Fourier transform of an image, giving you the amplitudes and phases of the Fourier components that you get in these 2D crystals, allowing us to get first a low resolution map, 7 angstroms in 1975, with a 3D model, and each of these 7 wooden rods are again 10 angstroms apart, and again are made up of 7 transmembrane alpha helices, the same ones that were seen by Pauling and Kendrew and myoglobin and so on. And after another 15 years between 1975 and 1990, we ended up with this reasonably good atomic model of bacteriodopsin, which you can see phenylalanine, tyrosine side chain, tryptophan, and then this is the beta ion ring of the retinal chromophore that gives it its color. So that was my entry into cryo-EM, and allowed me to appreciate what the power of electron diffraction is compared to other methods like X-ray diffraction or neutron diffraction. So now that's the kind of end of the general introduction. So we're now coming to single particle cryo-EM, which is the method that has made the big impact. And even until maybe 7 or 8 years ago, there were not many people doing single particle cryo-EM, but now it's become a major activity because of the success and the increased power of the method. So what were the major milestones that allowed single particle cryo-EM to develop into a powerful method? Well, one of the main ones was the development of a method of making these thin films of amorphous ice in which your protein molecule or your virus or your ribosome or your muscle proteins were embedded. And this was the plunge freezing method that Jacques Dubochet and his colleagues developed while they were at EMBL in the early 1980s. And I'll show you a couple of slides about that in a moment. But even in 1982, the microscopes were very primitive. They were not, they had tungsten filament sources, so they weren't very bright. The electron illumination was not very bright. So we needed field emission gun sources to be brought in. The ability to look at frozen specimens was limited by the technology of cold stages. So much better cold stages had to be developed and designed and all the problems eliminated. And then the last one was that the vacuums in those earlier microscopes were very poor. So as soon as you cooled the specimen, it would get covered with ice from the residual water vapor in the vacuum. So the gradual development of many aspects, stable microscope columns, electron optics, so that was a major, major important, and many, many people, including the companies, were involved in that development. And then in more recent years, the thing that's made it go from being a method with promise into a method that could deliver the goods were the development of better detectors. So prior to about 2010, the best way of collecting images in the electron microscope used photographic film, which you then had to develop, fix, scan, digitize, and so on. Phosphor-coated fiber optics that led to slow scan CCDs was an intermediate step, but the big thing were the introduction of direct electron detectors, which came around about 2011, 2012. Meanwhile, in the background, the power of computers and the software that various groups had developed was improving. And in the early days, Spider from Joachim Frank's group and Magic from Maren van Heel's group were the dominant technologies and computer software used in single particle approaches. But since about the year 2000, first EMAN with Stephen Ludke, Freeline with Niko Grigoriev, which is now called System, XMIP, developed by the group in Spain, and then Relion from Sean Sherry's group in Cambridge. These have all gradually been improved and now are very powerful methods. And then alongside this, the microscopes have also become more highly automated so that you can collect thousands of images in a day without being present. So you can go home, have dinner, have a beer, and meanwhile the microscope's collecting thousands of images. And the pioneering work was done with Leginon from Bridget Carricker and group in and Clint in at Scripps, Serial EM, and now the Mastronardi, and then EPU is the commercial version that FEI and now Thermo Fisher have installed on their microscopes. And so these have all been very important in getting the methods going. So now let's say a little bit about each of them. So here is Jacques Dubochet on the right, and here are two of his early apparatuses. This is images published in a very influential review they wrote in 1988 in Quarterly Reviews of Biophysics with his colleagues Alistair McDowell and Mark Adrian. And Alistair McDowell is the one who first plunge froze into liquid ethane at the temperature of liquid nitrogen. So the way it works is you put your little drop of your biological purified molecule ribosomes, viruses, on a little electron microscope grid, plunge it into a little container of liquid ethane at the temperature of liquid nitrogen. And these were early gravity-fed plunge freezing and elastic band-powered plunge freezing that was the basis. And now, of course, these are commercialized. You can buy them from Thermo Fisher, the Vitrobot, or Leica, or Gatan. So there are various commercial versions of this that work in a more automated way. So one of the earliest applications of the method done by Jacques' group was on the adenovirus particles, which are 100 megadaltons or so. And this was a picture published on the front cover of Nature in 1984. And you could already tell, just from looking at this picture, that the method had great promise. So you can see intact viruses. You can see some that have broken open, presumably during the plunge freeze process. You can see the occasional little black dot, which is one of the subunits that's fallen out. You can see the spikes from the adenovirus where it attaches to the cells and so on. And this was the state of the art in 1984, but none of the software, none of the methods for doing the analysis were really very well developed then. So it took from 1984 till 1991, so 6 or 7 years, to go from images in 2D like this to a 3D model like this. And this is from Phoebe Stewart's work whilst she was a postdoc at the EMBL, and shows that the structure of the adenovirus at 35 angstrom resolution. So it's you can see the images are fuzzy, the structure is fuzzy. It's good enough to see the subunit structure, but it's not good enough to see any of the higher resolution and the atoms that you might well be interested in eventually. And the reason for that is that the quality of the illumination, the quality of the detectors, the quality of the microscope stages was not adequate to give you. And we describe this with a B factor, a blurring factor, which describes the way that the different Fourier components of the structure fade as a function of resolution. And so this is a B factor of 3,000 square angstroms. That's a big fade out, and that's what's limiting the resolution to 35 angstroms. That's how it stayed for quite a long time until probably 20 years later. And here is another image of adenovirus taken in Hong Cho's group in Los Angeles, UCLA. And you can see now, instead of it being a somewhat fuzzy image of the virus, there's now a lot of internal structure inside the particle that shows that the image has a much higher resolution. And that's because the microscopes, the vacuum, the field emission gun sources, the stability of the stages have been greatly improved between 1985, 1990, and 2010. But the images are still, this image on the right here is still recorded on film before the detectors came in. Hong Chu and his colleagues processed images like this. This is the same image, got a structure like this, but now instead of at 35 angstroms resolution, at 3.6 angstroms, so 10 times better resolution. The B factor is 10 times lower. There's 1,000 times more data in this. So it's 10 times more data in terms of the resolution in 1 dimension, but 3 dimensions, it's 10 cubed. And that allowed them to get densities for the subunits that make up the adenovirus. Here you can see 3 strands of the beta structure in the particle. And you could even see the beginnings of side chains at 3.5 angstroms, a tyrosine and an arginine here on the bottom right. And that's where things stood in terms of the development of the microscope hardware technology until the new detectors came in. And this is a picture from the work in our group in Cambridge, evaluating 3 of these new direct electron detectors in 2014. The black line is what we call detective quantum efficiency of film as a function of the resolution in terms of the pixels after the film is digitized. And you can see it's about 1/3 DQE, 30% DQ. That tells you that of all the electrons arriving at the film, really you are only recording about 1/3 of them. And the signal to noise ratio in these images of biological structures unstained in ice made by the Dubourget method is very low. So small improvements make a big difference to the quality of the images. And so these 3 colored lines show you the 3 earliest of the direct electron detectors. The Falcon 2 then, it's now up to Falcon 4. The red curve was from work that we had done in Cambridge over many years, which became eventually the Thermo Fisher Falcon series of detectors. The blue line is the Gatan detector that was developed in California by Peter Denez's group with David Agard and Yifan Chen in the front line of testing it. And the reason it has a higher DQE at low resolution is that this was a very fast detector. So it could be used in counting mode, giving you a higher DQE at low spatial frequency. And then the green one was Direct Electron, which was another Californian company that developed a detector. And these are the three that are still the companies that deliver direct electron detectors. And with these, rather than with film, it had two advantages. One, it has a higher detected quantum efficiency. And secondly, the images come in the form of movies. So if there was any blurring of the image, which there was in the earlier ones on film, instead of it being recorded as one image that was blurred, you had a movie, maybe 20, 30, 40 frames that could be then subsequently processed in the computer to unblur the images. And that's the basis of the current technology. And so I'm just going to show you half a dozen images now of what the new detectors look like when you take images of real structures. So this is an image of pyruvate dehydrogenase complex, 1.5 megadaltons, taken by Vinod Kumar when he was a postdoc with us in Cambridge. He's now the leader of a group at Bangalore, leading the Indian electron cryomicroscopy facility there. And this was a grid made in the year 2000 by Peter Rosenthal. Of this, we worked on it and got it to about 8 or 9 angstrom resolution in 2003. But this image you can see now is very beautiful. This is the carbon film that you see bottom left. This is a hole in the carbon film. This is the thin film of ice blotted by the Dubochet method. So this is amorphous ice, and these are the icosahedral enzyme particles with 5-fold views, 2-fold views, and roughly 3-fold views, and then all the other orientations as well. And that allows you to get much higher resolution. And as you come down in size for the particles that you're looking at by cryo-EM, the images are just as nice, but there's less information because the particles are smaller. So the next one is a single particle cryo-EM image without any symmetry, which is mitochondrial inner membrane complex number 1, which consists of 45 different polypeptide chains in a membrane in detergent in these complexes. And again, this was image taken by Vinod Kumar in collaboration with Judy Hurst's group, and I'll show you this in more detail later on. And then coming down in size from 900 kilodaltons to half a megadalton. This is beta-galactosidase, the LAC-Z gene from the very first operon. It's got 4-fold symmetry. And if you look at some of these images, you can see here, for example, there are 4 little dots, which are the subunits of the beta-galactosidase. And then if you look around, you can see all the different orientations. So you can see by eye the orientation. And obviously in the computer, you can find out that much better. So then taking another jump, a factor of 2, this is an image of catalase. And here the blotting done by Dubochet method has squeezed out the molecules from the central region into a ring here where they're mostly in the same orientation, interacting with the air-water interface, and then further out, molecules in all different orientations, and right at the edge of the hole in the carbon, they're overlapped. And so it's the ones in the middle here, the images of the molecules in all orientations, that gives you the information, that gives you the 3D structure. Then coming down in size again, this is a 100 kilodalton structure, a pentraxin called CRP from Mark Pepys' group in London. It's a pentameric homopentamer, and you can see these little rings of 5 subunits, and the edge-on views as a little line here with 2 or 3 dots. And then coming down further, we come to hemoglobin, the work of Max Perutz using X-ray crystallography. And here you can see all the particles, but it's harder to decide whether you can find out the orientation or not. And when this image was taken around about 2015, we thought it was not possible to get structures at this size and smaller because there just wasn't enough information in the images of each particle to find the orientation. But as I'll show you a little bit later, that is now possible, and a student, Miriam Koshai in Martinsried, has determined the structure of hemoglobin from cryo images like this with a slightly different type of imaging. If you come down, one last image is 40 kilodaltons of albumin. And now, again, you see the little black dots are the particles, but now there's less information. And as you go smaller, for example, down to lysozyme or myoglobin, you can still see the particles, but at the moment, there isn't enough information. The quality of the images, the quality of the technology isn't good enough to get atomic structures from such small particles. So now I'm just going to show you what is the consequence of analyzing images like this using the current software. And I've picked a few examples that show the power of the method in its best view. So this is the first one. This is the work of Alexi Amunds, who is now a group leader in Stockholm, but was a postdoc with Venki Ramakrishnan when he did this work in Cambridge. It's bovine mitochondrial ribosomes. That was the goal of the work he was doing. And he spent a couple of years trying to purifying them, trying to crystallize them to use X-ray crystallography to determine the structures. But he could never get them completely purified because there's not many ribosomes in the mitochondria, and they were always contaminated with cytoplasmic ribosomes from the beef heart where they got it. So he collaborated with Xiaochen Bai in Shor-Sherez groups in Cambridge, and the two of them together took images like this, processed them with Shor-Sherez Rely-On software, and got an initial model of the mitochondrial ribosome like this. But one of the features of Rely-On is that you can then do either 2D or 3D classification of all the particles that went into giving you that structure. And along here you have the separated classified views, and the ones in the rectangular box are indeed the mitochondrial ribosomes they were interested in, but some of the other particles are cytoplasmic ribosomes or things they don't know what they are. But among the mitochondrial ribosomes, there's intact ribosomes, but there are also partly degraded ones where you only see the large mitochondrial subunit here. So there's, and so these can then be selected from the entire data set and used by focusing in terms of finding the orientation on either the large subunit or the small subunit of the ribosomes to give you structures like the ones in the top right here, where by selecting the particles that are only mitochondrial ribosomes and then focusing in terms of orientation determination on the large subunit, you can get a structure for the protein molecules and the RNA in the large subunit, and then separately the same data but a different computer procedure focusing on the small subunit and get atomic structure of the entire ribosome. And so within a fairly short period of time, I think most of the work was building the models because there's such a lot of new proteins. They were able to get a map with features like this. So this is 2 or 3 regions from that map. A little bit of Watson-Crick RNA double helix on the left with a resolution of about 3 angstrom resolution, showing the stacking of the base pairs and confirming that the resolution that the program tells you you've got is reflected in the quality of the density map. And then this is a top view of the same Watson-Crick base pair, an alpha helix with side chains, and 2 beta loops, beta turns with side chains. And this is what's thought to be a magnesium ion at one of the turning points. So that showed you a lot of the features of cryo-EM that make it so powerful. The ability to work with samples that are not completely pure, the ability to focus on one region of a structure that's fairly flexible and then extract the structure of the whole thing. So a second example comes from work of Xiaochen Bai and Shor-Sherez in collaboration with a group at Tsinghua, Yigong Xi's group, working on gamma secretase. This is a membrane protein that's thought to be involved in producing the beta amyloids that may underlie Alzheimer's disease. And many people had worked on this membrane enzyme because it's a drug target, possibly to decrease the incidence of the beta amyloid deposits in Alzheimer's. But no one had been able to crystallize it. And until this work was done, there were no structures beyond about 20 angstrom resolution. And the image doesn't look all that great, but when you do 2D classification, you can see the membrane domain and the hydrophilic domain sticking out from it. And by putting those together and creating a 3D model, they eventually got a resolution of 3.4 angstroms. And here at top left, you can see it's 4 subunits, one hydrophilic subunit, nicastrin, and then 4 membrane domains of the 4 different subunits in different colors here. So presenilin, nicastrin, and APH1, and these are then the densities for them have been plucked out and showed individually. And you can see again there are 20 transmembrane helices, and they were able to find, identify, and build into the density the amino acid sequence of the protein, showing you just as good structure now as you would get from X-ray crystallography. As well as the alpha helices, there are regions of beta structure, particularly in the nicastrin, and these are some stretches of beta structure. That's the one single green transmembrane alpha helix. It's the part of the nicastrin molecule. And then on the surface, there is glycosylation. So here are 3 sugar clusters that are connected to the glycosylation region of the nicastrin protein. And even in the membrane domain, a couple of reasonably intact looking lipid molecules with the side chain, with the lipid fatty acid tails. So again, this was a structure that was impossible to be done by any other method. So a third one comes back to the mitochondrial complex 1 that I showed you. This is from the work in Judy Hurst's group in another MRC unit in Cambridge. And the electron microscopy was done by Vinoth Kumar, as I mentioned, back now in Bangalore. And he took images like these back in 2014 and processed them to give you this initially 5 angstrom structure of each of these single particles without symmetry. And this is Shaipen Ju, who's back in China. Judy's still the group leader in the MRC mitochondrial biology unit in Cambridge, and that's actually the head of it at the moment. The map shows that at a high contour level, there are these 8 brown iron sulfur complexes in a string, and the way this enzyme works is that an electron from NADH gets fed in at the top. It then hops its way down the iron sulfur complex and then reduces a quinone in the membrane. And here there are 76 transmembrane alpha helices, the same ones that Linus Pauling had discovered. And at this stage, they weren't really able to see 5 angstrom resolution, what the interpretation of it was. But you can see first the detergent and lipid belt that surrounds the alpha helical part. And in this, eventually an atomic model done 2 years later, you can see that the polypeptide's been colored in 2 colors. The blue are the core subunits, of which there are 16 polypeptides, and these had been in a homologous structure determined previously by X-ray crystallography from Leo Sassanov's group. And Leo is now in Austria as a group leader, having switched back from being in X-ray crystallography to doing cryo-EM also. But the red ones are the additional 29 what they call supernumerary subunits that are not present in bacterial homologs. So the work of Leo Sassanov was a thermophilic bacterial version of mitochondrial complex 1, about half the size, about half a megadalton. But in the mitochondrial membrane, you've got all 46 polypeptide chains, nearly a megadalton in size with no symmetry. And that was able to be determined from images of the type that I showed you. And they were able to build models and identify all the different subunits. And both Judy Hurst and Leo now have done a lot more work. And other groups in Germany and China are also working on this. Okay, so those were 3 structures that show different facets of the power of electron cryo microscopy. And I'm going to just show you 3 others that show you how cryo-EM has been being used in other types of work other than bigger complexes. So the first one is this work from Miriam Koshai, who was a PhD student with Radiston Danef. Danef is now in Tokyo. Miriam Koshai now works for a pharmaceutical company in Switzerland. But this is the work she did as a PhD student. And she thought she would try and use the faceplate that had been developed by Radiston Danef on hemoglobin. And when she took these images, like the one on top left, both her supervisor, Radiston, and her head of department, Wolfgang Baumeister, told her not to do this experiment. It wouldn't work. But she ignored them and did it anyway, and took images like this over one weekend, did the 2D classification using Relion, got some structure, and eventually, by putting it into Relion, they eventually got the structure of the whole hemoglobin, 64 kilodalton, 4 polypeptides, 4 heme groups. And here's the heme group. Here's the proximal and the distal histidine. And that is essentially the same structure that you saw back in the Irving Geist structure of the atomic structure of myoglobin, but it's now in hemoglobin. And so that showed you that the range of the cryo method is not only for big structures with multiple subunits or with and without symmetry, but it can also be done on smaller subunits. And so here are a couple more. This is some work that Vinoth Kumar and Giuseppe Canoni did in collaboration with Rams, who's now in Purdue, but was in Bangalore when this is phenylacetic acid degrading enzyme. It's a trimer, and these are the structures of 3 different complexes with difference maps shown in red of the different ligands that are part of the enzyme mechanism, and they're enlarged on the bottom. So octenyl-CoA and NADPH are 2 of the ligands that are involved in the reaction to degrade the phenylacetic acid in this enzyme structure. And so this shows that not only can you determine structures, but you can also do difference maps and look at ligands, and you can look at enzyme mechanisms and how they work. So this is a good example of it, particularly because it's got substrates and cofactors that are big with clear structure. But you can also use it on structures like this, and this is again using a faceplate. Javier Garcia Nafria, who's now back in Spain, he was a postdoc with Chris Tate in Cambridge working on GPCRs, and this is his image on the top left. This is his 2D classes. This is the Fourier shell correlation showing a resolution of 3.6 angstroms. This is the structure of the this is serotonin GPCR with the trimeric G protein bound to it. And if you look in this red square at the top and magnify it, you can see the density for the agonist donitriptan. And so with structures like this, the pharmaceutical drug companies can now begin to design better drugs to both activate or inhibit pharmaceutically important drug targets like GPCRs. Okay, so that's the end of some examples. And now I'm going to try and sort of wrap up with some generalizations. So here is a picture of the statistics from the Protein Data Bank of structural biology's contribution to sort of world knowledge in the area over the years by the 3 methods using X-ray crystallography, electron microscopy, and NMR. And you can see that in 1997, so 20 years ago, there were really no atomic models being deposited based on electron microscopy, sort of 1, 0, and 1. Whereas every year there were 1,000 or 2,000 structures done by making crystals, going to synchrotrons, collecting data, publishing the structure. And then NMR at this stage, you can see 300, about a quarter or a third of as many structures as by crystallography. And then as time went on, more synchrotrons were built, detectors at synchrotrons became better, and the X-ray crystallography has grown and grown and grown, so that now there are about 10,000 new structures being published each year using X-ray crystallography. And again, in the red curve, you can see the NMR grew and grew and grew until it peaked in 2007, and then this has declined now. And now there are very few de novo structures being determined by NMR. It's still being used for other things like low affinity ligand binding. But the growth of electron cryomicroscopy, you can see now, is up to, and I've extrapolated this year to be about 2,000 depositions. It's still about 5 times less than X-ray crystallography. So the statistic we like to look at in trying to predict the demand and what is needed in the different structural biology methods, we like to look at the ratio of the structures being deposited by cryo-EM to X-ray crystallography, and that's shown in this graph. And you can see, so in the year 2000, each year before that, there were probably 1,000 times more protein data bank atomic coordinate depositions by X-ray crystallography than by cryo-EM. It dropped with the advent of the microscope hardware to about 100, and then in roughly 2012, it changed to have this new trend because the direct electron detectors came in here, and now there's been exponential growth. Doubling time is roughly 2 years, and so if you draw a line through that, you can see that it's predicted that there should be roughly the same amount of structures done by cryo-EM and done by X-ray crystallography at synchrotrons in about 4 years from now. So it'll be, this 5-to-1 ratio should drop to 2.5-to-1 in 2022, and then it'll be 1-to-1. And then after that, it's a bit far to predict, but it looks like not only will it be cryo-EM responsible for determining structures of things that cannot be done by other methods, but it may also become the dominant method just for any structure that you're interested in. Okay, so now a couple of minutes left. Looking forward, what are the things that we're all interested in now? Well, one is we want to improve the methodology that people have been using to determine all these great structures. So bigger and faster detectors with a higher DQE, better faceplates would be good. There's some new work coming out with Chris Russo and his student Katerina Naidanova about new grids that help to reduce beam-induced motion. And then looking ahead, we're still all using liquid nitrogen, but it's possible that lower temperatures might be better. And from the work of Fujiyoshi, that suggests that's possible. But one of the things that we've been focusing on at LMB in Cambridge is to try to make, now that we know what we're doing and we know which of the developments in electron cryo microscopy really mattered and which didn't matter, although we did think so at the time, the idea is to make cryo-EM now less expensive so that you can do it in smaller universities with smaller groups with less money, rather than focusing on big labs with lots of higher level support. Okay, so just to demonstrate this, just a couple of slides on this last thing. So one of them is from work of Matthew Peat in Chris Russo's group at the LMB, and we decided that we would look into whether the electron microscopes we were using at 300 kilovolts were really the best electron energy. And there's lots of historical work that suggests that lower energy gives you more interaction of the electrons with the specimen, but there's elastic interactions and there are inelastic interactions. And the elastic scattering cross-section of electrons by protein structures gives you the information you need to get the image, to get the structure. The inelastic interactions do damage to the protein, cause radiation damage, and limits the exposure you can do. And it turns out the inelastic, that sigma I, is that orange curve, and the elastic cross-section vary with the electron energy, but these two curves are not parallel. And you can see they get closer together as you go to lower energy, and that says you get more information for the same amount of radiation damage by going to lower energy. And so we've explored this region between the two dotted lines, 300 kV, which is current state-of-the-art microscopes, and 100 kV, which is cheaper to make. And you can see that if you look at the ratio of elastic to inelastic, there is a slight advantage as you go to lower energy, although you lose some electrons. And so what we've got in these dashed lines is that for different thicknesses of the specimens, you can plot the number of electrons you've got left times the ratio of elastic, and you can show what is the optimal energy. So for 300 angstrom thick specimens, it looks like 100 kV is best, but for thicker things, 300 kV is best. And so that was the theory behind what we're trying to do with these lower energy microscopes. And then the second slide is the practice. And so this is Katerina Najdanova, this is Matthew Peat, and others. And this is an image, one of a half a dozen structures we looked at, and this paper is published about last October in ultramicroscopy, and it shows the structure of DPS, which is a 200 kilodalton structure with 2-3 symmetry at about 3.4 angstrom resolution. And you can see the alpha helix. This is an alpha helix in that structure. So this shows that in theory and in practice, we ought to be able to do better by developing some of the technology involved at 100 kV. So now I'm going to stop by having a brief thing. What further improvements can we expect? There are various things that we can be doing, both for thin specimens for single particle work and for thicker specimens for tomography. And there's still factors of perhaps 10 or 20 to be gained. I won't go into that because I'm now 1 or 2 minutes over time. Instead, I'll just do the acknowledgements. So I showed you a selection of cryo-EM structures from our work in Cambridge and from other people's work. Adenovirus, mitochondrial ribosome, Alexei Amunts, gamma secretase, Xiaoxuan Bai, who's now in Dallas as a group leader, associate professor there, doing lots of great work on ion channels and other structures. Mitochondrial complex 1, Vinos in India, Sharpens in China, and Judy still works on complex 1 by EM, Miriam Koshai, and then these other structures, and the work on detectors in Cambridge. Greg McMullen, Wazi Faruqi, the two people that I've worked with, Renato Torchetta and Nicola Guarini, who were originally at Rutherford Appleton Lab, and Renato now has his own company in Barcelona, and all of them are continuing to develop better detectors, which we need for the future. And these were the people involved in that 100 kV project. So with that, I think I should stop and hand back to Valentine. So let me know if I have succeeded in finishing, and I will come out of that and try and get back to my screen.
Thank you for the great talk. Now we have some questions from the audience. And the most upvoted question is from Panos. The question is, how plausible is to introduce time scale in cryo-EM structure for molecules that show motion?
Yeah, okay, so now that we have all these powerful methods, of course, all the different people in the field are taking different tacks on what they should do. So many people are very keen to look at mechanisms and look at time-resolved reactions as they proceed. And one of the troubles is, of course, you only get the great images if you freeze the specimen and do it by cryo-EM, and it's frozen. So nothing can happen once you've plunge frozen the specimen, put it into the electron microscope. You determine the structure of what you have at the point of freezing. So anything to do with looking at changing structures and mechanisms has to be done in a separate prior step. And of course, the plunge freezers, normally you blot the sample and plunge freeze it, but you can do various preparation methods prior to that plunge freezing. And there are a number of groups, for example, in Leeds, in the UK, they have something between 10 and 100 millisecond plunge freezing apparatus. In New York, the group of Bridget Carricker and Clint Potter, they've developed Spot It On, which again can give you quite good time resolution. And then originally Nigel Unwin's work involving spraying on. So Nigel's work back in the early 1990s on the acetylcholine receptor involved spraying 1 molar acetylcholine in little droplets onto a grid. As the grid was plunging through the air into the liquid ethane, just before it hits the ethane, he would spray it. And with that, you can get resolutions of 5 or 10 milliseconds. And so there are a variety of groups tackling a variety of projects. Another one would be, I know Joachim Frank in New York have done ribosome assembly studies on the kinetics of when you mix together the different components of ribosomes, simulating how they're being synthesized in the cell. You can follow the kinetics over times of minutes by plunge freezing and then analyzing the spectrum of different structures that you get. So there are things you can do, but it's all very piecemeal at the moment. And I don't think anybody has found any way of doing the cryo-EM other than by the plunge freezing. So it's always going to be a method of doing kinetics, triggering the reaction, and then trapping different intermediates at different stages.
Okay, thank you. There is another question from Julio. Is where do you see X-ray crystallography, electron crystallography, and cryo-EM standing in the next 10, 20 years?
Yes. Okay. So we are all interested in this, of course, because it tells you, if you're a company manufacturing equipment, it tells you where to put your effort in developing new equipment. If you're building a university or a research institute department, it tells you what sort of people to train, where to put your money, and so on. So it's terribly important. So at the moment, it's still the case that once you have a purified protein, if you can crystallize it, it's far quicker to determine the structure by X-ray crystallography. So most of the pharmaceutical companies, there are 30 or 40 of them that do structure-based drug design. If you have a good crystal of your drug target, your protein, your enzyme of interest, and you want to screen thousands of drugs, you can do 300 or 400 a day at a synchrotron. Whereas in the cryo-EM, you can probably do 1 a day or 2 a day. So at the moment, once you have a crystal, X-ray crystallography is far quicker, which is why you've still got 10,000 structures of PDB deposition per year, but only a couple of thousand done by cryo-EM. But there's a capacity problem, so the cryo-EM will get better. I don't think the crystallography will ever go. There's such a lot of investment has been done in synchrotrons. They'll never go away because they're used for lots of other things other than structural biology. So that will continue. But there are many structures now that are impossible to do using crystallography. So I think our view is that, or my view certainly is that cryo-EM is already in many areas the dominant technology. It'll become more so. And so our thought is that maybe in 10 years, there'll be lots more microscopes, there'll be lots more structures per year. And exactly what we will be doing in 10 years using 3D X-ray crystallography, it's not clear to me, because by 10 years from now, we will know. You just look up, if you want to know a structure, you'll be able to look it up in the database. It'll already have been, we're already at the stage where you know about half the structures in biology, or have homologous models for them. But there will still be, for example, let's say there's another virus like the coronavirus. You will need rapidly to determine the structures of drug targets in that virus, perhaps antibody complexes and so on. So there'll be an ongoing need to invent new reagents, to determine the structures, to develop better drugs. And so probably in 10 years, we won't be worrying too much about how you do it. You'll be focusing the power of the methods, which will be X-ray crystallography at synchrotrons and cryo-EM with possibly more sophisticated 100 kV microscopes, or faster and better 300 kV microscopes. And then there'll be other structural methods like tomography that will come in that you won't be able to do by any other method. And then the third thing you mentioned, the electron crystallography, which was the way I made my transition from being an X-ray crystallography to being an electron cryo-microscopist. The difficulty there is you still have to make a crystal, even though it's a 2D crystal. And, it's not necessarily any easier to make a 2D than a 3D crystal. So whereas it had there's probably 10 or 12 atomic structures done by electron crystallography 2D crystals, there are not many people doing this anymore. We've stopped about 1995. Henning Stahlberg is probably the leader in doing this, but he also does single particle cryo-EM. And also, if you take an image of a 2D crystal and it's not terribly well ordered, you can use the single particle methods to process the images of the 2D crystals. And similarly, people working on helical structures now, they actually use single particle methods to process as the images of their helical structures. So it's the single particle methodology that's beginning to dominate all types of, and even in electron cryotomography, where you get the tomogram, eventually people are cutting out subtomogram 3D regions, and you're using single particle subtomogram methods to determine higher resolution structures of repeating objects that are in that tomogram. So I would say, it's going to be the single particle style of approach, whether it's from tomograms or from individual crystals or from individual molecules, that will become dominant.
Thank you. So the next question is from me. What is the major limitation of cryo-EM resolution now, and how to overcome this limitation?
Yes. Okay. So when we were involved in trying to develop cryo-EM to be better in the 1990s, we were very happy with 10 or 20 angstrom resolution. And then Bettina Boccia was the first one to go below 10 angstroms with single particles. And then Hong Cho with adenovirus, for example, on film. And then gradually, and then I have to say, my own view is that 2 or 3 angstrom resolution is adequate for almost all the questions in biology that you're interested in. And if you remember, back in 1959, the first protein structure of myoglobin was at 2 angstrom resolution. That was the first structure. But as you went from 1959 to 1960 to 1970, people realized that you could build perfectly good models because you only have 20 amino acids and 4 bases with structures that were not at 2 angstrom resolution. So I remember we backed off 2.5, 2.8, 3, and then gradually, as the structures people wanted to know became more difficult to work on, the resolution actually got worse using X-ray crystallography. And eventually, people were determining atomic models with 4 and 5 angstrom resolution maps. So you only need 2 angstrom resolution if you want to do pharmacology, 3 angstroms if you want to do find the relationship of your structure to other structures or to other intermediates. However, there's nothing to stop electron microscopy going to 1 angstrom or half an angstrom resolution. So there are perfectly good half angstrom resolution structures of metals or in material science. And there are now a number of protein structures at about 1.2, 1.3 angstrom resolution done by cryo-EM. So actually, there isn't really any limit. You can go on if you take enough particle images, but you have to ask how many structures are there that are going to tell you something interesting between 1 angstrom and 2 angstroms, and I don't think there's so many. So I think what we need is a lot more investment to make it easier, quicker, and less expensive to do structures between 2 and 3 angstroms, and then a few people who wish to push the methodology should be free to try harder to push the resolution limit. And so I think it is true that people quite like the idea of saying, my structure has the higher resolution than your structure. So this is all to do with, your personal predisposition. What is it you like to do? Do you like to work on one or two structures and keep pushing it, finding out more and more subtle features of the structure? Or do you want to focus on some new greenfield area of biology that hasn't really been tackled by anybody so far, in which case you don't need such high resolution?
Okay, thank you. And the next question is from Carmen. What suggestions would you give to tackle pseudosymmetry except for symmetry expansion and focused refinement?
Yeah, so many structures are, let's say, homodimers, but it turns out that the two halves of the dimer that are related by a twofold aren't exactly related. So you can often get a slightly better density map by processing the single particle images using no symmetry, so that's C1 symmetry, rather than C2 symmetry, which is twofold symmetry. And so I think a typical approach is that usually with a structure, you have some idea what you're expecting to start with. For example, if it has a molecular weight of 4 times the polypeptide, you know it's a fourfold symmetry of some kind, but you may not know whether it's 2-2 symmetry or C4 symmetry. And so what isn't so well developed at the moment in the single particle cryo-EM software is a sort of semi-automatic method to allow the experimenter to find out what is the true symmetry of their structure. So when we were working on two-dimensional crystals, in the same way that single particles, you have cyclic symmetry, so Cn, you have dihedral symmetry, D1, D2, Dn, you have tetrahedral, octahedral, and icosahedral. So there are the 5 single particle symmetries. In 2D crystals, there are 14 plane groups that allow proteins to have a structure. And so because people often had a difficulty, they took an image of a crystal, they got spots, but they didn't know what symmetry, what the space group was. So we have a program that we wrote called AllSpace, and it simply tests out all the different symmetries of the 2D crystals that you can get against one image that you get. And then it tells you what symmetry best fits your observations. And at the moment, we haven't really got... so usually what happens is people collect a data set, let's say that's 50,000 particles, and then you simply say, I would like my software, so rely on system, EMAN or whatever, I would like to process it using the symmetry I guess. And then you just try a few different symmetries and then you look at it by eye. So what would be nice would be if somebody would develop an automatic procedure. You say, I don't know my symmetry program. Please, can you just try everything you can think of and then tell me what the result is by giving you some statistics at the end? And that's what our 2D crystal program did, but it did it from one image. Whereas now you obviously has to, that would take, a few days of computing time, but that would be a useful development. Whereas at the moment, you just have to do it, and then you look at the structure and you ask, is my map improved? So I think it's a little bit piecemeal, a little bit of trial and error at the moment. And there are a number of forums. There's 3DEM, there's CCPEM, and problems like this are, if you have a particular problem with a particular set of data, you can put it up there and you'll get dozens of suggestions from people who have experienced similar questions with different structures. And then you can distill from that maybe how you should proceed. But there isn't at the moment any automated way of doing it. And maybe that's one of the things that the software developers might focus on next.
Okay, thank you. And probably the last question for today's session is from Julio. What is a major bottleneck for obtaining high-resolution structures from heterogeneous samples? the technology or the sample preparation?
Yes, so there are, now that we have all these methods, of course, there are lots of people that focus on using it for different things. So one interesting idea that involves looking at, let's say, impure specimens, one idea was you don't need to bother purifying your structure at all. You could just take a whole cell, smash it up, put the whole thing on a grid, and then you'll see all the molecules there. And I have to say, we once tried, I thought, well, it would be nice to get some pictures of hemoglobin. So we just took a drop of blood, lysed the cells, and put it all on the grid. And you can see the hemoglobin, but you can see all the other enzymes, catalase and so on, that are in the cytoplasm. And at the moment, the images and the processing methods are not powerful enough to sort out the mixture of molecules that you get. So I noticed one idea was you lyse the cell, you have the entire contents of the cytoplasm, or if you dissolve it in detergent, you have all the different membrane proteins. You run that through a size exclusion column so that you get the big molecules, the medium molecules, the small molecules, and then you can make a grid using size exclusion sorted mixtures of structures and try to extract from that by 2D and 3D classification the structures of the molecules. And you don't know what they are initially, but you'll recognize some of them probably. So there are people trying to do that. And I think eventually, once let's say the detectors are a bit better, this beam-induced motion that I haven't talked about gets completely cured, you'll be then having a method where the images and the computer programs are almost perfect and you cannot improve them. And at that point, you may well be able to, with enough data, determine hundreds of structures just from one data set. And that would be one possible direction for development, looking at, and of course you can also say, okay, I tried my best to purify the thing I'm interested in, and I could spend another year getting it better, or why don't we just look at it as it is? And you may well be able to sort out that there's only 3 or 4 other things, and you'll be able to determine the structure of your main topic of interest alongside other structures that are impurities. And this is not dissimilar to what some of the X-ray crystallographers experienced. They were often trying to determine the structure of something, a membrane protein. They'd make a crystal, but when they analyzed it, they'd find it was a crystal not of their target, but it would be a crystal of a 1% impurity that just crystallized much more easily than the thing they were trying to crystallize. And for example, the structure of fatty acid synthetase, a lot of those X-ray crystal structures were done by people working on ribosomes, because fatty acid synthetase and ribosomes were about the same size. And when they would get crystals, they would find it wasn't the ribosome they were interested in, but it was a different molecule. And so they said, oh, well, we've got a crystal, we might as well determine the structure, even though they weren't really interested in it. So there are parallels in other areas like that, from X-ray crystallography into cryo-EM. Yeah, okay.
Okay, thank you, Dr. Henderson, for awesome talk and very interesting Q&A session. And today we also have a panel discussion at 5 p.m. And the next talk is given by John Michael Petters. And see you there.
Thank you very much. Yeah.